In previous lessons, we've learned about the importance of the cell nucleus. It is necessary for survival and reproduction of the cell. In Hammerling's work, we learned a little bit more about what the nucleus is actually doing in the cell. It is producing information molecules that move up into the cytoplasm of the cell, and these information molecules are called messenger RNA. Messenger RNA molecules are temporary recipes to build proteins. Here we see the red squiggles would be messenger RNA molecules, and they are recipes to build proteins, which would be the blue dots. So in his work on the algae that can regenerate, the regenerated cap would be constructed from proteins that are built according to the recipes of these messenger RNA molecules. Now in this lesson, we'll inquire as to the origin of these messenger RNA molecules. They're coming from the nucleus, but what's inside the nucleus? We will learn there are structures called chromosomes, and chromosomes are composed in part of DNA. So we'll begin to think of messenger RNA molecules as temporary copies of information stored in DNA. In this lesson, then, we'll learn about these structures called chromosomes. Now, we mentioned that the nucleus is important for survival and reproduction. Those are two different things. The survival part here can be seen in this diagram, that the nucleus is important for survival because it's making the recipes to build proteins, and proteins are the workers of the cell. To see how the nucleus is important for reproduction, let's consider again Hammerling's famous two-cut experiment. You'll recall that the foot piece will regenerate a cap, but surprisingly the stalk piece will also regenerate a cap, and the cap piece dies. Now notice that was surprising because it didn't have a nucleus, and we came to an understanding why the stalk has the power to regenerate. It has accumulated messenger RNA molecules from the nucleus that it was attached to. So the stalk has messenger RNA from the nucleus, which are used to build proteins that can regenerate a cap. But notice the stalk does not have the information to build another nucleus. You see, we never saw that stalk piece ever regenerate another foot with a nucleus. Now that's an important observation. So the information to build another nucleus must be residing in the nucleus itself. And the nucleus, we will learn, contains these structures called chromosomes. So if the cell is going to reproduce, then reproduction of a cell will require the duplication of the nucleus, which means all the structures inside the nucleus, these chromosomes, are also going to have to be duplicated. So while the survival of the cell requires the nucleus to be making messenger RNA molecules to build proteins, the reproduction of the cell requires that the cell duplicate the chromosomes, the permanent recipes for building proteins, so that two daughter cells can have all the information needed to make proteins. Now, let's take a look at what these chromosomes are. If you take a look at the right side of the diagram here, we see a molecule of DNA. Now, DNA is technically called a double helix. It's like a twisted ladder, and we'll examine the structure of this molecule in more detail later. But we see here that a chromosome is composed of a very long molecule of DNA, and then there are these spheres here. These are proteins called histones, and we can think of them as packaging proteins. This really long DNA molecule is going to wrap around these histones and coil up even more into coils and supercoils until we have a really compact structure. And the compacted structure of DNA wrapped around proteins is called a chromosome. Now, as we continue our study of reproduction and heredity, we're going to be identifying DNA as the molecule of heredity. It's the molecule that has those three properties. It has the capacity to store information, it can be duplicated, and the information can change, thus accounting for evolutionary change. So if we go back to the acetabularia, then chromosomes are within the nucleus. Chromosomes are composed of DNA molecules wrapped around proteins, but it's the DNA molecules that will have the recipes for building proteins. And messenger RNA molecules are temporary copies of the recipe to be delivered out to the cytoplasm where protein construction takes place. Now, chromosomes are visible with a light microscope, but they're only visible in the compacted coiled state. when the DNA molecule is sort of unwound here from its packaging proteins, the DNA molecule is too small to see with the light microscope. Electron microscopes can see it. Chromosomes can look like this. These are salivary gland cells of a fruit fly, and here we see a bunch of chromosomes sort of taking the form of the nucleus. A cell has been kind of squashed, the nuclear membrane has been squashed away, and here we see chromosomes in the form of the nucleus. There are four cells here would have been squashed. Here, if you squash the cell a little bit more, you can spread out these chromosomes and see something like this under the light microscope. So these are chromosomes of a fruit fly. These are the structures then, the hereditary structures that are found in the nucleus of the cell. They are the permanent recipes for proteins, and they will be copied in temporary copies that we call messenger RNA. In this diagram, then, we'll look at the relationship between chromosomes, messenger RNA, and proteins. So here we are in the nucleus of a paramecium. So the whole thing here is our paramecium. We have a food vacuole over here. We have the nucleus of the cell here. Inside the nucleus are these structures called chromosomes. And chromosomes are composed of DNA wrapped around proteins. But here we see a section of DNA. Now, we'll be learning that section of DNA that has the recipe to make one protein is called a gene. In this case, we have the gene for a digestive protein. Now, when the cell needs to make a digestive protein, it's going to copy the information of that gene into a molecule of messenger RNA. That messenger RNA molecule then will leave the nucleus. This is Hammerling's information molecule coming out here, and it will hook up to a factory, a protein construction factory. It's called a ribosome. And the protein construction factory will assemble the protein, in this case a digestive protein, according to the recipe in the messenger RNA. And the messenger RNA is a copy of the information in DNA that we call a gene. So the molecule is DNA, but the information contained in that molecule is called a gene. Well, in order to properly do digestion, of course, you remember you need digestive proteins to break down the food, but then you need transport proteins to get the nutrient molecules out of the food vacuole. You're going to need that's a different protein. It has a different shape. It has a different job. And so there must be a different recipe to build that protein. Sure enough, here's that recipe hooked up to the factory, building the transport protein. And that recipe, that temporary recipe in messenger RNA, originated from a different gene. So here's the recipe for the transport protein. So every protein in the cell has its corresponding gene, which is the recipe to make the protein. And the first step in making a protein is to make a temporary copy of the recipe in the form of messenger RNA that then leaves the nucleus and hooks up to the ribosome, the protein construction factory. Now remember, these messenger RNAs are temporary, so they will degrade over time. But that's not a problem for the cell because the cell can just make new ones. If new proteins are needed, the cell will turn on the gene, will start using the genes to make new proteins. Now you'll notice in this diagram that when a cell has to build a protein, it's going to be accessing the information of DNA in order to build a temporary copy. Now we haven't figured out how that happens yet, but we will in a later lesson. But nevertheless, the information has to be accessible in order to make a messenger RNA molecule. Now let's return to that other diagram. We're going to make a distinction here. When the DNA molecule is unwrapped from these packaging proteins, that's precisely when the information in DNA is accessible and messenger RNA molecules can be built. On the other hand, if the DNA molecule is wrapped up around these proteins and coiled and supercoiled into this structure over here on the left, then we say the DNA information is not accessible. Now, why would a cell make its information not accessible? Well, this process of coiling up will be one way to regulate DNA activity. If you just wrap up a section of DNA, then you're going to turn off that gene. The cell will not make that protein. In order to make a protein, then you're going to have to unwrap that recipe for the protein so it is accessible. But another reason that DNA is going to coil up is when the cell is going to reproduce, when the cell is going to divide into two cells. And we'll see that process in the next lesson.